Yeah, hi everyone. So today I'm actually filling in for Kristen Agosta, who unfortunately couldn't present today. So she was going to talk about small mammals and some really interesting results she had from her honours work. So I'm just the backup fill-in. I'm no Kristen, but hopefully next year she'll be able to present her honours research. So today I'm actually going to be talking about a project that Jess mentioned briefly in her talk. And that's the Wild Otways rewilding component. And we're working on the swamp Antechinus. So this talk's not going to have any data. This project only started in June this year or beginning of July. So I'm just going to talk through what we're planning, what we're thinking about doing. And if anyone's got any feedback, we'd really welcome that. So I'll just mention before I start that I'm working on the lands of Wurundjeri today, and I'd like to pay my respects to their elders past, present and emerging. So as just briefly mentioned, the Australian government has funded the Wild Otways initiative over 3 years, and this is kind of the last year of funding. So our project is funded for 12 months. And we're working under this rewilding sub-project, which was about designing and creating a rewilding program for threatened small mammal species in the Otways. So before I came on board in July, this program had an element of expert elicitation where they undertook a big process of going through all the species, all the small mammals that were present in the Otways, and they assessed these against things like habitat suitability, the different threats that were in the ecosystems, the availability of different small mammal species for translocation, whether they had captive or wild populations, as well as any negative effects that species could have on each other, so things like competition and predation. So these things were all ranked against each other and clustered, and there was a final report that was produced. And out of that, the species that came out ranked as a potential species for translocation was the swamp Antechinus. So following this, the CMA contacted Zoos Victoria along with Barb Wilson due to expertise on previous translocations and expertise with Antechinus ecology and biology. So that's how this project kind of came about. So here's our lovely little swamp Antechinus, our target species, and we'll be working on the mainland swamp Antechinus. There's also a subspecies in Tasmania as well. So this map here just shows all the records, and it's actually been a bit cut off, but the species extends from all the way in the east near Wilson's Prom, across through the Otways and further west all the way to the South Australia border and into South Australia a bit as well. And you can see that a lot of these records are really close to the coast, so it occurs only up to about 200 metres elevation. So just a bit about their biology. Swamp Antechinus are a carnivorous dasyurid, so they're in the same group as quolls and Tasmanian devils. They live in really wet habitat types, so wet heathland, sedgelands, coastal scrubs and tussock grasslands. And they have a carnivorous diet. So they spend a lot of time digging around in the soil looking for insects, so the larvae of moths and beetles. They'll also feed on small vertebrates and sometimes seeds as well. What's really fascinating about Antechinus is their amazing reproductive strategies. So they have this really synchronous reproductive period in winter that just goes for a couple of weeks, and the females and males will breed during that time. But once that couple of weeks is over, all the males in the population will die after mating, leaving just the females behind. So sometime between June and August, the female will give birth to a litter of up to 8 young, 6 to 8 normally. And you can see this image up here. It's a female that we caught a few weeks ago. She'd probably just given birth quite recently. And she has tiny little pouch young in her pouch that are about the size of a grain of rice. But she'll carry them for quite a long time, as you can see from this bottom image, until they're about 2 months of age. They'll be still hanging on as the female runs around looking for food. And after that stage, she'll pop them in a nest that she's built, and she'll come back and forwards while she's still lactating as they spend another month or so in the nest. So it's a pretty cool little system. The species is listed as vulnerable under EPBC, and it's been declining for a number of years. One thing about Antechinus and this mating system is that they've evolved under a really stable environment. So this makes them really susceptible to extreme events, particularly things like prolonged droughts. So as you can imagine, given that they only breed once a year, they just have that one litter. If something happens to that litter, the female can't breed again. There's no more males in the population. There's no chance of having another litter until the following year. And that's if she survives. So populations can really sort of go up and down quite quickly. They're also facing a number of other threats. So things like predators, foxes and cats, habitat loss, degradation of habitat from grazing, both from introduced introduced herbivores and native herbivores, changing fire regimes, and other impacts of climate change, such as reduced rainfall or heat events. Another presumed threat is the lack of gene flow between populations. So some of these swamp Antechinus populations are really fragmented across the landscape. And so there's probably not much gene flow going on between different populations, but that's something that we're interested in exploring more in this project. So as conservationists, we don't have all that many tools, but one tool we do have that we can trial is things like reintroduction. And that seeks to restore viable populations of native species within their former ranges. And so that's something that this project is about, exploring whether or not that's an option for this species, and collecting a lot of genetic samples to look at what's going on with the genetics of these isolated populations. So I'm just going to talk through a little bit about what we're planning. And as I said, this is all sort of like very early days. We've only been doing this work for a couple of weeks. And yeah, if there's any feedback, very welcome to provide it to us. And a lot of this work might change over time as we get new information and new data that's going to guide what we decide to do. So we're currently in stage 1. We've been starting our planning and spending a bit of time securing all our ethics and permits to undertake survey work and monitoring of this species. We've been doing consultation with traditional owners, and we've been working with Barb Wilson on her small mammal projects, and we've been getting out and about doing some live trapping surveys over the past sort of six or 7 weeks. So we've been out trying to catch Antechinus at sites where Barb and Mark had camera trap images of the species to try to get tissue samples from live animals that we can get analysed for their genetic diversity in an analysis that's going to occur between September and December. So we're working with some partners at the University of Sydney, and we're going to send those samples off to them for genetic analysis to see what's going on. We've also been talking to a lot of other researchers. So we've contacted the Melbourne Museum and researchers from Deakin and the Nature Glenelg Trust, who have worked on swamp Antechinus in the past as well. And Kristen, who was going to present today, also has about 60 samples from her honours work. So far we've already got almost 100 samples that we're going to go and get analysed later this year. And then we'll undertake some camera surveys while females are lactating and leaving their young in the den. We'll do some further monitoring using cameras rather than live trapping. In stage 2, we want to do some additional work. So we'd like to do some habitat analysis, look at sites where the species is present, look at the habitat there and maybe compare that to sites where the species used to be, where they're no longer present, see what's going on with the habitat. We'd also like to look at predators and other threats in the landscape at sites where the species used to occur but now no longer occurs. And then we'll commence some of our translocation planning. So as I said, this is going to be greatly informed by the genetics analysis that we undertake. And we'd like to do a lot of site selection, thinking about things like the data that we have, where we know they exist, where they used to exist, but we'll incorporate other things like cultural burns and the threats in the landscape when thinking about which sites we might source animals from and which sites we might release animals into. Part of this work will also include population viability modelling and a disease risk assessment. So all of this stuff has to be done ahead of any translocation work that might happen. And no translocation can just happen immediately. It involves a fair bit of paperwork. So there'll be applications made to the Translocation Evaluation Panel, which sits with DELP. So we'll get feedback from them, and we'll put in new ethics applications and permits to undertake this additional work if it's deemed necessary. So prior to any translocation occurring, things that we need to think about are stuff like site preparation. So again, as I mentioned, looking at the predators, threats and disease analysis. And we're thinking about some site modifications that we might be able to trial, working with other people as well. So things like these wildlife tunnels that have been installed by Deakin and trialled post-fire, whether or not they're suitable for something like this. Whether remote weigh stations can be used and trialled in the field, whether microchip feeders could be of use for this species, and obviously cameras will be used in monitoring post-release as well. And then if all of this work comes together and a translocation does occur, animals will be captured using live trapping methods, and we target a translocation to the dispersal period. So between April and June, a lot of males are moving around the landscape looking for females. So we'd kind of work in with the biology of the species and trial anything around that time. Once animals were captured, they'd be transported to new sites. There might be an element of supplementary feeding that occurs, and then we'd really want to monitor them quite intensively to look at survival, body condition and health parameters. But yeah, this is something that was trialled many years ago by Barb Wilson. So there's a bit of data that we already have about what works and what doesn't work. So we'd just be building on that as a team to look at alternative methods. So finally, just to finish up, I'm not sure how I'm going for time. As Jess mentioned at the beginning, there was a little bit of funding from that emergency bucket of money that has just been secured by Barb and has come through to us to run a little fire project in the Carlisle Heath. So we know from previous work that Barb's done on small mammals that swamp Antechinus are quite late at recolonising habitat post-fire. So in some cases, it took longer than 15 years after the Ash Wednesday fires for swamp Antechinus to turn up at sites again. So too frequent burning in the landscape could be an issue for this species, but it's possible that cooler or patchy burns might be okay. So we're looking to address the question, at what age post-fire does vegetation become reoccupied by swamp Antechinus? So this is a pretty small project. We're just focused in the Carlisle Heath. We want to assess what our current data that we have. We've got camera and trapping data, what we have, and compare it with fire history. Kristen, who is going to be presenting today, has great data from her honours project in the Carlisle, so we'll be able to use a lot of that, and some of those sites have since been burnt, so we'll hopefully be able to go back and trap at some of her sites that she trapped at last year. And she also showed from her honours work that medium to long intervals since fire supported higher abundances of mammals at her site. So we want to look at this a bit further. But this is all really early days. We haven't even really had a chance to work out how we're going to do this project yet. So all we know is that we'll do some more planning, and we're going to get out there in November to do 3 weeks of trapping and habitat assessments at sites. So yeah, I think that's basically it from me. I'd really like to thank all of our partners. This has been a really amazing collective team effort. Particularly like to thank Barb for all of her data and sharing all of that with us, because we've been able to launch into things quite quickly, because we had a really good solid base of small mammal data across this landscape to get started with. So it's been fantastic. Thank you.
Thanks, Gemma. You've got time for one question. So
Great.
In relation to genetic analysis, this is from Melanie. Is this based on live captured animal samples or also utilising hair traps or other indirect survey techniques?
Yeah, great question. It's all based on tissue samples at the moment. So we have access to some from Kristen's work, some from the museum, and some that we've collected from live trapping. So we're not planning on using any hair or anything like that at the moment. We seem to have enough tissue samples that we can get our hands on for now. So yeah, thanks for the question.
Thanks, Gemma. We're out of time, but there's a couple more in the Q&A. If you wouldn't mind having a go at answering those and posting them, that'd be great.